Now I'm thrilled to introduce this year's Berger lecturer, Eric Pierce. Eric is known to many of you as he served on our faculty from 1999 to 2011. In fact, he sat right next to me. After completing his PhD in Madison, then MD, residency, and pediatric ophthalmology fellowship at Harvard, he worked alongside Jeff, both at Harvard and here at Penn. He then returned to Harvard, where he's now the William Shatlos Professor of Ophthalmology. His extremely productive career, recognized recently with the Arvo Proctor Medal, has been focused on genetics and therapies for inherited retinal degenerations. He's had numerous NIH grants and served as chair of the PED1 study section. He's published 127 papers, including one last year with Tomas in New England Journal of Medicine on gene editing in patients with retinal degeneration. Eric, please come up and receive the Berger Lecture plaque. So Eric's talk is going to be entitled Genetics of and Genetic Therapies for Inherited Retinal Degenerations.
Thanks, Josh, for this lovely introduction, and thank you all for the invitation. It's a real honor to be here to give this talk in Jeff's memory, and also a great pleasure to be back to be able to visit with all of you here at Penn. It's impressive what's happened here since I left in 2011. The department has obviously grown, and the whole medical center looks like it's really flourishing, which is fantastic. As Josh mentioned, I overlapped with Jeff when we were residents at Mass Pioneer, and I got the help of the archivist at the Mass Pioneer Library to track down a photo to try to prove that we were actually there together. And it turns out that we were. It's hard to tell, but that's me in the background back there, and Jeff hiding here. And in case you don't believe me, I blew them up a little bit. So again, me kind of hiding and Jeff kind of hiding. Obviously, we shared some sense of wanting to be in the back row. But that really wasn't Jeff's personality. There's one incident that I'll relate. I remember after I just got here when Jeff came over to the FM Kirby Center to welcome me and kind of show me around a little bit. And we walked across the bridge from the Stella Trans building to what was then called BRB1. I don't know what the current name is. And as we're walking through the hall, there's a beautiful sort of atrium there that's used for functions, and we're walking along that atrium. And Jeff pulled me over to one side and introduced me to somebody that I hadn't known. And it turned out it was Glenn Galton, who at that point was the vice dean for research. And I don't know about the rest of you, but I get pretty intimidated when I meet those kinds of authority figures. But Jeff was not intimidated at all. He walked right up and said, "Hey, Glenn, it's great to see you. I really want to introduce our new colleague, Eric Pierce." We chatted for a while. It was a wonderful introduction. And when I was reflecting on that incident and others like it, I kind of come back to what Josh said, which is Jeff treated all of us like family. He was very warm in that regard, and his warmth is what carried through that encounter. But he was also very self-confident. And I respect that and still kind of envy that many years later. So it's a real pleasure and honor to give this lecture in his memory. As I was thinking about what I was going to talk about, I actually updated my title and the content a little bit, because again, as Josh said, Jeff was really at the forefront of using digital analyses of retinal images to study and track disease. And what he was really doing was identifying imaging biomarkers of macular degeneration that could be used to follow patients and predict their outcome. I think he would have been in love with the current rise of machine learning-based and AI-based tools for these kinds of analyses. So since he was looking at biomarkers, I thought I would tell the story we're trying to develop on biomarkers as well. I'm not an imaging specialist, so I'm sticking to my strength, which is molecular biomarkers, and I'll get to that in the second half of the talk. Just here are my disclosures. Spring has finally made it to the Boston area, so the cherry trees are in bloom, and the Patriots are out to fight tyranny. When I left here in 2011, I left because I was recruited to Harvard to set up what's called the Ocular Genomics Institute, or OGI. This is an institute, a translational research institute, where we study the genetics of inherited eye diseases, how the mutations or variants in genes we find lead to disease, and try to use that information to develop genetically informed therapies. It's thrilling that that is happening in our field, as Josh alluded to, and we're going to hear throughout the day today. I'm very excited that I think that OGI is now a successful endeavor at Harvard these 14 years later. There are 15 full-time faculty associated with primarily associated with the Institute, several other team leaders, and it's really a wonderful group to work with. The OGI team studies multiple different kinds of inherited eye disease, both rare inherited forms of disease like retinal degenerations, which I'm going to tell you about, and also common complex disorders like glaucoma and other optic neuropathies and age-related macular degeneration. My area of interest, as you heard, is inherited retinal degenerations. I know you're very familiar with these because of the wonderful groups here that study these disorders, focused primarily on the Center for Hereditary Retinal Degeneration with Artur and Tomas leading that effort, with Gus's effort for the animal work, and of course with the long-standing contribution and continued presence of Sam Jacobson in that regard. So you know that these are disorders that cause vision loss by damaging the photoreceptors and retinal pigment epithelial cells in the retina. And by losing those cells, which you can see on the OCT here, where the cells outside the fovea are missing, that causes reduced visual field. We'd like to be able to intervene to be able to prevent progression of that to total loss of photoreceptor cells and total loss of vision. This is challenging because these are very genetically heterogeneous disorders with over 280 different disease genes identified to date. What we're trying to depict in this crazy Venn diagram is that the little symbols are the symbols of all the disease genes identified for inherited retinal degenerations to date, and they're grouped, or we attempted to group them by their phenotypes. So you can see, for example, for CEP290, it's in the LCA, or severe early onset retinal degeneration bubble, but it can also be associated with more adult onset rod cone degeneration, or RP, or ciliopathy. That kind of crossing and overlapping of phenotypes makes me want to suggest that we call these disorders by their genetic names when we can, and include the phenotype information as well. So if we know a patient has CEP290 associated LCA, let's call it that. And I think we need to move away from the kind of historic eponyms that we've used in the past. Having this many disease genes makes it challenging to develop therapies, but fortunately that we're making great progress towards that goal, as Josh suggested, based in a large part on the pioneering work done here, led by Jean Bennett and Al McGuire, to develop the first gene therapy for inherited retinal degenerations, which was also the first gene therapy approved for any inherited disorder in 2017. There's a lot of progress in that direction. In order to make that progress, in order to bring these therapies to patients, we have to understand the genetic causes of inherited retinal diseases for the patients we see. So if you sequence all the exons of those disease genes I showed you, a test we developed just as I was leaving Penn, you can find the genetic cause of disease in about 55% of patients. Our research, which I'm not going to talk about today, but I just want to mention, is now really focused on finding the genetic cause in the remaining 45%. And the short story is, as I think you could predict, that if you start looking outside the exons of those disease genes into the non-coding portions of the gene, you find a significant contribution of genetic causality, probably in a quarter of patients, due to non-coding variants that alter splicing or structural variants in the genome. And we're working hard now to bring that testing to clinical use as well. But I'm not going to talk about the genetics. I'm going to talk about therapeutics and biomarkers instead, if I keep my time right. And I forgot, of course, to start my timer, so I will do that now. I don't see a clock, but I'll use this one. And that's not what I wanted, but we'll give it a go. As I said, gene therapies are very promising for treating, or genetic therapies very promising for treating these kinds of inherited retinal diseases. Most of that work is AAV-based gene augmentation therapy, following the work of Jean Bennett and Al McGuire. For genes that are too big to fit in AAV, or have different mechanisms of disease, like dominant disorders where gene augmentation won't work, other modalities like gene or base or prime editing are being deployed, or antisense oligos. And fortunately, we're going to hear about all of these during the day with what I'm going to tell you now, with Tomas and Artur's and Peter Quinn's talks throughout the day. The one key feature here is when to do a successful clinical trial, the drug not only has to work, but you have to collect data that supports an approvable clinical endpoint. And the magic for the Luxturna story is that in the lunchroom in the FM Kirby Center with materials from Home Depot, Jean Bennett and colleagues developed this multiluminance mobility test, which is really a walking full field stimulus test. It's a way to detect the sensitivity of the retina and the photoreceptors to light. Instead of pushing a button like in an FST test, patients get to walk through the maze. But it's a walking, I would call it a walking FST. And after treatments, patients' visual sensitivity improved dramatically, and that's been sustained. That kind of endpoint is really important because, as we'll get into, while I think a lot of drugs that are currently being tested work well, they're not getting approved because they're not meeting their pre-specified clinical trial endpoints. So endpoints, I think, really become a critical feature in our field, and I think that's where the biomarker story becomes critical, as I'll get to. As I said, there are multiple clinical trials of these genetic therapies in progress. I've tried to list all the ones that I know about here. I'm thrilled to work with Rachel Huckfeld and Jason Commander in the OGI at Mass Eye and Ear on our inherited retinal disorder service, where Rachel is leading a number of trials with Jason as the vitreoretinal surgeon who does the subretinal injections. I got to lead one trial, which is for CEP290-associated retinal degeneration, which I'll tell you about in the next few minutes. And that starts, as it does for many of us who see patients, with our clinical experience. Sorry, again, I didn't get that quite right, so let's just go back to a clock. There you go. When I was at CHOP across the street, although I'm not sure I could figure out how to get there today, and I guess that we're not seeing patients there anymore, they're in a different location. I met a young infant named PD whose family brought her in because she couldn't see. Typical severe early onset retinal degeneration, what we call LCA. Nystagmus, hemorrhagic pupils, and couldn't respond to the parents' facial expressions. We've been following PD since then. She's currently in college and doing remarkably well as a musician with her light perception, vision, and nystagmus. But we've always had a discussion with that family when they come to see us, is there any way that there could be treatment that could improve this young woman's vision? And we hope that that's the case because, as you can see here, even though her vision is light perception, there are photoreceptor cells in the fovea in this OCT image. That's structure function dissociation, as Sam Jacobson so clearly defined and helped us understand. So could we treat those remaining photoreceptor cells, get them to work better again, and improve vision? When we did genetic testing for PD, we found that she had CEP290-associated disease, as I mentioned, including a common deep intronic variant, which activates a cryptic exon and it activates the mutant allele. And that's a great target for all kinds of genetic therapies, and particularly we think genome editing, which I'll tell you about. That, as I said, that variant, deep in intron 26, activates a cryptic exon, which causes a null allele for CEP290. If you could somehow contrive to prevent that by, for example, using genome editing to remove that mutation, or as I think you'll hear about later, antisense oligos to suppress that abnormal splicing, you could hopefully restore expression, at least in part, of the normal protein. In about 2014, so 10, 11 years ago, a new company in Boston, Editas, approached us about trying to figure out what would be a good target for some of the early studies of in vivo genome editing. Which could we try in patients? They were attracted to eye disease for many reasons. And we and many others around the world suggested that they consider this mutation. And they did. They decided to use CRISPR-Cas9 gene editing to treat that mutation. This is kind of version 1 CRISPR-Cas9 gene editing, where the Cas9 directed to the spots in the genome makes double-strand breaks. We thought the intron was a good place for that to occur. There are many additional genome editing technologies. I think you'll hear about some of them today, like base or prime editing, that could be used in modern versions of this and are being deployed now for treating inherited retinal diseases and other inherited disorders. So Editas, the company, developed their drug EDIT-101, which is an AAV that encodes a Cas9 nuclease and 2 guide RNAs that flank this deep intronic variant and hopefully excise it, restoring CEP290 expression. Editas did a fantastic job with preclinical development of this. They showed that the EDIT-101 drug corrected splicing in cultured cells, restored protein expression in cultured cells and in organoid models. They did an amazing job testing for off-target activity, because you can imagine one of the risks of genome editing is you could edit the wrong place. One of the reasons this was such an attractive place to start is the intron 26 variant is surrounded by thousands of base pairs of intronic sequence, so there's a lot of room to test guide pairs for excising this variant. Editas tested thousands of them and found a pair that has high efficiency of cutting and no reproducible off-target activity. So I think that set the standard. Morgan Mader, who did that work, I think really set the standard for how to approach this for clinical development. While that preclinical work was being done, Editas sponsored a natural history study looking at potential clinical endpoint outcomes. Endpoints are so important, as I mentioned. And of course, the group at the Center for Hereditary Retinal Degenerations here have been doing the same thing for much longer. Based on that, the preclinical work and the natural history study, a clinical trial was planned. It's the standard dose escalation study of 3 adult cohorts with increasing dose, leading to 2, hopefully, pediatric cohorts at what were determined to be safe doses. The primary endpoint here is obviously safety, but 4 efficacy outcomes, acuity, FST, patient-reported outcomes, and a new maze that was developed, a new walking test, were chosen based on the natural history studies, plural. When this started, this was the first clinical trial to ever use CRISPR-Cas9 gene editing in vivo, period. So when the first patients were treated in this trial, no one else in the world had ever been treated with gene editing in vivo. Since then, there have been multiple studies that have been done in vivo editing that have reported results, and we're thrilled that the Brilliance trial is one of those with the New England Journal paper that Josh mentioned earlier. So keep that in mind as we go through the study results. This is the first in human use of a brand new technology. So think about how you might judge that if you're thinking about success. The data I'm going to present is not the full data to now. The Editas has provided data for the 14 subjects who were enrolled in the trial, 12 adults and 2 children before they suspended enrollment, which we can talk about. The longest, as you can see, is 2 years, the shortest 6 months, so about a year in average follow-up. So it's early clinical data. We're working, we hope, on trying to collect more follow-up data at present. So what did we learn in this study? The most important thing is EDIT-101 has a favorable safety profile. There were adverse events, of course, but most of them were mild or moderate and mainly related to the surgery required to deliver the subretinal injection. There were 5 adverse events of special interest. There were no serious adverse events or dose-limiting toxicities. There were 5 adverse events of special interest, most of those due to the appearance of subretinal deposits on OCT, which are thought to be inflammation. In some subjects, those resolved spontaneously without any treatment and without any disruption of vision. As I'll show you when we get to the efficacy data, there was one subject who had reduced vision while she had those subretinal deposits and required steroid treatment. Here's the efficacy data, years of work in one slide. What it shows is the efficacy data change from baseline for the 4 main outcomes: FST, best corrected acuity, the maze performance, and the patient-reported outcomes. And the dashed lines were pre-specified thresholds for clinically meaningful improvement. And you can see we've organized the chart based on the results of the FST, because 6 subjects had clinically meaningful improvement in their threshold, in their ability to detect light, and in this case, red light. So it's red FST, so it's cone function, which makes sense given that we're talking about restoring function of foveal photoreceptors. So 6 of 14 improved in FST. 2 additional subjects had improvements in macular vision with acuity. 4 subjects had improvement in the maze test. I wish we had stuck to the test that was made at the FM Kirby Center. This one was much more complicated. It took hours for patients to perform. Tomas is smiling because he remembers. Not a great idea. It became, I think, a little too variable. And the patient-reported outcome measures here, 4 subjects improved in that as well. But that was the NEIV of Q25. There are better patient-reported outcome measures for inherited retinal disorders that have been developed since, but they weren't available when we started this study. Just to give you a little flavor of what the patients say, they thought this helped, many of them thought this helped them. They could see the food on their plate. Their worst seeing eye, which was treated, had now become their better seeing eye, where their visual fields were improved. The same kinds of things we heard when we first started the Luxturna trials here at CHOP in 2007. Here's the patient PD, who I introduced to you, who had improvement in her threshold in her ability to detect red light, and she thought that improved her quality of life. It's not all perfect. As you can see, there are a couple of subjects who had some decreases in vision. This is the subject who had decreased vision while the subretinal deposits were present. That patient has left the study after starting a family, but is followed remotely by an outside ophthalmologist, and we know that her vision is at least partially recovered as those deposits resolve. Tomas and Albert McGuire did a fabulous job recruiting the 2 pediatric subjects in the trial here at CHOP and following them. And Tomas demonstrated to us that the pediatric subject has some of the largest improvements in their photoreceptor function. And indeed, the subjects who started out with the worst sensitivities had the largest improvements in their retinal function. So I think these data support the path to a potential phase 3 clinical trial and an approved drug. Editas decided to suspend enrollment primarily for business reasons. We can discuss that if I can make sure I leave time for questions and answers. I'm about 15 minutes in, is that correct? 23 total left. Perfect, thank you. Great. But if we're going to move this towards a phase 3 clinical trial, we have to have an approvable endpoint. I think FST could be that based on what the data we've accumulated in our field, but we have to convince the FDA of that as well. So I think the gene editing study here, this Brilliance trial, was a success. Again, remember, first in human ever use of a new technology, turned out to be safe. There's early evidence of in vivo editing occurring because the patients improved photoreceptor function. So I think this serves really as a proof of concept that we can use these kinds of gene editing techniques to treat eye diseases and other inherited disorders as well. We need to make sure to acknowledge the participants in the study. Those of you who've done this kind of clinical trial work know it's an enormous undertaking for subjects to participate, our patients to participate in these trials. It takes a lot of time, and of course they're undertaking significant risk as well. I do also want to thank Editas. I think they did a wonderful job. They were a great partner sponsor as we did the study, and we're working with them now to try to move this program back into clinical development. Hopefully we can achieve that. All right, change gears and try to finish up this part of the talk in maybe 10 or 12 minutes to leave time for questions. We'd like to deploy gene editing techniques for other diseases, as are a number of you I know. One of them is Usher-2A-associated retinal degeneration, which is a common cause of not only Usher syndrome, but non-syndromic retinal disease. And as you can see, a lot of mutations are clustered in one exon, exon 13. And the goal of all this work is to skip exon 13 with antisense oligo or with gene editing so that we can restore function. It turns out that the protein produced from a gene that doesn't have exon 13 functions both in preserving hearing and retinal structure. But there's a challenge here. This and many other inherited retinal disorders progress slowly. Looking at data again from the wonderful team here at Penn, you can see that for changes in visual field to occur, either light adapted or dark adapted fields in patients with Usher-2A-associated disease, it takes decades. It would take 3 or 4 years for a detectable change to occur if you compared a treated eye that didn't progress any further with an untreated eye that continued normal progression. So that time of separation of those two curves, 3 or 4 years, is too long for most companies to support for clinical trials. They want a faster outcome. So as we've dealt with this in our field, we've actually hit a few trials not meeting their primary endpoints. And I think that's partly because in some cases, maybe the drug doesn't work as well as we hope. But I think mainly it's because we didn't have time to get to the right clinical trial endpoint before the sponsors assessed whether they wanted to continue or not, which I think is what happened for the Brilliance trial, at least in my personal experience. As we followed patients longer, we're seeing there are continued benefits that accrue, but you need time to do that. So as we've talked with pharma and biotech companies about this, what they've said is what you need is a biomarker that could tell us if there's target engagement, did the drug work, did it make its biologic effect in the retina before the change in vision might occur? Did we correct the defect in the rods or cones and they're happier and healthier? So now we can have the confidence to wait to see the natural progression separate from that preservation. So what's the right biomarker? There are people in our field doing all kinds of studies for this. Jeff was working on imaging biomarkers, as I mentioned. My strength is molecular ones. So we're trying a different kind of biomarker, which is extracellular vesicles as biomarker. And I think you're probably all familiar with this based on Jeff Sundstrom's wonderful work in the area. What Jeff and his colleagues showed years ago is that the vitreous is chock full of these extracellular vesicles. They're small lipid-bound vesicles that are produced by all cells, and they contain the contents of the cells they made them, like RNA or protein, or in some cases DNA. They circulate, they move within the intercellular space in organs, and they also reach the systemic circulation and circulate. And because they contain the contents of the cells that made them, they're being used as biomarkers in multiple fields, cancer being a big one. As you can see from this drawing, there are EV biomarkers being used for studying all different kinds of cancer. Most of them are RNAs, and in particular microRNAs, that are used to see if a patient still has cancer or if metastasis is present. So maybe I'm being simple-minded, but I'm thinking if you can detect a metastasis by looking at the contents of the extracellular vesicles that produces and sends into the circulation, and a metastasis is a small thing to start at least, why can't we do that for our eyes? Why can't we find the vesicles made by our eyes that are in the systemic circulation and use their contents to assess retinal health? So that's the hypothesis we're trying to test. We're not the only ones doing this. Others, as I mentioned, Jeff's group is certainly working hard on this and making great progress. Others are using aqueous or vitreous to check for ocular tumors or glaucoma, and are starting to look at circulating biomarkers made by the retina for things like melanoma and maybe AMD. So we're trying to do this for inherited retinal degenerations. And we decided, rather than take on the task of looking in the systemic circulation first, we wanted to start with a simple system. We could ask, for the retina, does it hold true that the EVs made by retinal cells, like photoreceptors or RPE cells, reflect what's happening? Can we detect RPE degeneration, for example, in an RPE culture by looking at the vesicles? So we picked a simple story. For years, we've been studying what we call RNA splicing factor, RP, mutations in a group of genes that encode components of the spliceosome are now actually the most common cause of dominant inherited retinal degeneration. We don't understand why. That's what we're studying. How come if you disrupt components of the spliceosome, you get non-syndromic retinal degeneration? It's a good question. One thing we learned, actually starting here when I was at the FM Kirby Center, was that at least part of the way that that occurs is by disrupting RPE function. There's a cell autonomous defect in RPE function in the RPE if you have a mutation, for example, in the PRPF31 gene, which is the most commonly mutated gene among these. What you see, for example, is RPE from iPS-derived RPE cells that have a mutation in PRPF31 have funny shapes, they don't have good barrier properties, and they phagocytose poorly. So we thought that's a great simple model to start testing the idea that the EVs made from these cells might reflect RPE degeneration. Heron Getachew, a postdoc, took this on with the help of Marcela Gurita Hernandez, who's our IPS expert. It takes 4 months to make mature RPE from IPS cells. But Heron did that for 12 biologic replicates of wild-type and PRPF31 mutant RPE cells. She collected, after they were mature, she collected conditioned media from them, isolated the EVs, isolated the RNA, and studied the RNAs. And I'll just quickly go through this. The EVs in the mutant and the wild type are the same. We collect both large and small EVs because we're happy with any that turns out to be a useful biomarker. What's kind of cool is that you can see that the EVs in this Venn diagram contain almost all the RNAs, this is poly-A RNAs, that are expressed in the parent RPE cells, which we harvested at the end of the experiment. The EVs also contain other RNAs. A lot of them are regulatory, like link RNAs. If you compare the RNA contents of the EVs from the mutant cells with those out of the wild-type cells, about 500 EVs RNAs, poly-A RNAs, are highly enriched in the mutant cells. The enrichment here is 1 million to 1 billion fold. That's kind of cool. And there are about 300 poly-A RNAs that are depleted to a similar degree. There are also microRNAs that are enriched or depleted, a little fewer, 12 and 6. Just very quickly looking at the microRNA story, What's cool of the 18 microRNAs that are enriched or depleted in the mutant EVs, a group of them are associated with RPE dysfunction and retinal degeneration. And associated here means the genes they regulate are involved in retinal function and disease, like RDH11, which is in the retinoid cycle. So I think we're seeing biologically relevant changes in the RNA contents of the extracellular vesicles from the RPE cells. Can we extend this? And I think I'll just quickly finish up. We're doing this in animal models of disease, which is kind of cool. We're doing this now in human retinal organoids because we want to see if the retina has the same story. Afiz Beg, a postdoc on the group, has shown that retinal organoids made from these PRPF31 mutant iPS cells are also defective. This is, the green staining here is rhodopsin staining rods in this laminated organoid, and there's obviously much less of that in the mutant organoids. Now we can look at those vesicles. We want to extend this to animal models, taking advantage of a really nice genetic tool where you can make membrane-bound vesicles turn colors based on Cre expression. Pre-Cre in this mTMG mouse, everything's red. In tissues that express Cre, everything turns green, and this is in the membrane. So vesicles made from those retinas are green. You can sort them by nanofax and study what's in them. Very helpful. I'm not going to go through those data in the interest of time. I'm going to jump to the finishing story here.
You do have 12 more minutes to talk and then questions.
12 more minutes to talk, and how much for questions?
Then 5 minutes for questions.
So maybe 7 more minutes. Okay, then I will. Thank you.
12 minutes for you to talk.
I'm going to try to leave 10 minutes for questions. So 7 more minutes to talk. Thank you. I'm sorry. I usually manage my phone a little better. All right, I'll tell you briefly the story then. Thank you, Josh. That's great. So just very briefly, we thought we could use this model to help us figure out, to define the transcriptome, if you will, of the retina-derived and RPE-derived EVs, because you can sort out the green vesicles in circulation. We got some surprising and pretty cool results. When Heron sorted the control mTMG mice that have no green, right? There's no Cre. She didn't see any green signal. But when she sorted mice that are mTMG that are exposed to 6,3 Cre, which is a Cre driver that expresses in all retinal cells, she found a lot of green signal, somewhere between 5 and 10%. Now, we've gotten a lot of skepticism about this result because everybody says the eye cannot contribute 5 or 10% of the circulating extracellular vesicles. Maybe it doesn't, but these are the data. Let's at least have a chance at seeing if that holds up and if we can use it. We know this Cre isn't perfect. There's a little expression of the Cre in the somatosensory cortex of the brain, just a little. But to be really careful, we're making mice that don't have eyes. We can cross into this line. We can really define what's specifically present in these derived from the retina. So that's in progress. If you, again, look at what's enriched in the EVs that are green in this model, you find a lot of poly-A RNAs that are highly enriched, and you also find microRNAs that are enriched. So again, even in vivo, I think we have the capacity and the technology to continue to test this hypothesis. What I think is really cool is that we've started doing this in patients, a little bit as a just proof of concept. Can we get even close? So we collected plasma from 4 patients with severe retinal degeneration and 4 age-matched, in many cases, family member controls. We isolated the EVs from the plasma. We isolated the RNAs from the EVs, and we sequenced the poly-A RNAs that were present in those patient-derived EVs. And if you compare the EV contents, the poly-A RNAs, in the patient samples compared to the control samples, we found about 900 poly-A RNAs that are depleted in the patient samples. Half of those, about 400, we know are expressed in the retina, and a few dozen, some of them indicated by the gene symbols here, are retina-specific expressed genes, genes that aren't expressed elsewhere. So we're detecting changes in their expression and their presence in extracellular vesicles from patients with retinal degeneration. I think that's pretty promising preliminary data that we can detect changes in retina-derived EVs that are in the circulation in response to retinal degeneration. There's a lot more work that needs to be done here. One simple thing we're starting to collect samples for is we need kind of the in-between state right? We don't just need the extremes of the severe degeneration and the wild type. We need to know if someone's in the middle of degeneration, can we detect similar kinds of changes? We've written this grant a couple of times now, and our colleagues in study section remain pretty skeptical, but I'm hoping that at least you are willing to consider the idea that maybe a biomarker of early response to therapy or a biomarker of the degree of retinal degeneration could be, if we're lucky, a liquid biopsy. Just like we do liquid biopsies for cancer. You've probably all heard that term. Maybe we'll get lucky and have a liquid biopsy for retinal health. At least that's our hope. Because I think if we did, and we could test our patients that we treated with a genetic therapy 2 or 3 months after treatment and show they were responding biologically, there'd be much more confidence to do the trials in such a way and for a long enough duration that we could really find out if our therapies work effectively or not. All right, enough of that high horse. I think I'll finish up. I hope I've convinced you that genetic therapies are very promising for inherited retinal disorders and that biomarkers like perhaps EVRNAs could be really useful in helping us succeed with the translation of these genetic therapies to patients that we care for. I've tried to thank some of the folks who've done the work along the way, and at the beginning, I need to thank the whole wonderful group of investigators at the Ocular Genomics Institute that I get to work with. And as one final shameless plug, if you're at all interested, the International Symposium for Retinal Degenerations are great meetings. RD 2025 will be in Prague in September. Please consider coming. It's a wonderful meeting. Thanks very much. Yeah, I like that idea. We think we're pretty agnostic to where you get the vesicles from. Urine apparently has a lot of them, and they're kind of pre-filtered. So that's another potential source. We've been trying to think of something that'd be the least invasive. So I don't go right away to the periorbital sampling, but that's a very intriguing idea. Could there be a higher concentration in those vessels? If the story continues to develop in a positive direction, those kinds of things would certainly be worth evaluating. Absolutely.
And following on that, what do you think of the relative sensitivity of sampling EVs from the serum versus aqueous or vitreous sample?
I mean, I think there's no question getting an aqueous or vitreous sample would be more enriched in vesicles that come from the retina or other ocular tissues. And Jeff has done a great job not only studying these, but also developing an instrument that can be used to facilitate or simplify vitreous sampling. So it's possible we'll wind up moving in that direction. I was a pediatric surgeon, ophthalmic surgeon. I never felt that comfortable hitting the posterior segment, so I'm a little hesitant now to do those kinds of invasive procedures. But absolutely, if we can safely do those kinds of sampling, then that would be a great source of EVs.
Claire.
Yeah, Eric, that's fabulous work. And the idea of these biomarkers being predictive is a powerful idea. Do you know whether the EVs that you see in circulation are transported across the basolateral membrane? Have you looked at the polarity of their secretion across the RPE?
You know, we don't. We do grow our mature RPE on transwalls, so in theory we could separately sample basolateral versus or the two sides of the membrane if we wanted. I don't know if it's a perfect separation.
Yeah.
When we've done it so far, we've collected and pooled.
Okay. It's just interesting that there may be different EVs going in different regions for signaling mechanisms. So,
is there a reason you think the apical secretion might be different than basolateral?
there seems to be different polarity, and a couple of people have found that the EV patterns are different, and just the problems and what they may be communicating may be affected this way. I was just curious whether you're looking in circulation at a particular subset.
We could definitely determine that for sure, and obviously worth doing. understand
Anyway,
it better.
fabulous. A great study.
Thank you very much. You're very kind.
Tomas.
Hi, Eric. That's a wonderful job. I used to be a skeptic. I'm not anymore. I was wondering if you had officially or unofficially approached, regulatory people about how they would feel about us using EVs as a readout in a clinical trial. As you know, we struggle all the time to get approval for the things that matter to us the most. So I wonder if you had approached them.
So I have not discussed the idea of using EV biomarkers with any regulatory, not people knowledgeable about regulation and FDA regulations. But I'm not thinking about the EVs as a biomarker, as an approvable endpoint. What I'm thinking about it is as a way to assess what we've learned is called target engagement. Could we sample the EVs made by the participants in the Brilliance trial that we've both worked on so hard and tell if the gene editing took place because the cones are feeling better? So that we had the confidence to wait for the long enough to get the visual endpoints. I still think the endpoints need to be visual function based.
Really tantalizing data with respect to the differences that you see between normal retina and severely degenerated retina. Is the difference in the EV arising from the whole scale decimation of the retina, or is it arising from a lack of the expression of a gene? But I see the KCNB2, for example, is one of the genes that are there. Was wondering about your thoughts regarding patients who have a loss of function in those genes, whether that, even in the absence of wholesale decimation, you can still perceive the difference, because that suggests that the dynamic range they're looking for is actually present, and with gene replacement, you can actually follow the diminishing of those differences.
Yeah, well, I love that idea. It would be fantastic if we could collect vesicles from genetically defined populations of patients with inherited retinal disorders and then look to see if the affected gene or its partners in function have altered levels of RNAs and EVs. Absolutely. That would require a lot of patience. The grant we've written to support the human work here is a little more circumspect. We've figured out it takes just under 100 people in each category to have enough statistical power to have a significant result with just a 20% change in RNA levels. So if we could eventually get to that kind of number, absolutely, I'd love to be able to do that.
But we see KCNB2 being low in patients and someone who's not had very severe disease. That would really be very exciting.
Absolutely. Yes, we should definitely look at individual genes, and happy to take suggestions of which ones to look at for sure.
Gus.
Thanks, Josh. Beautiful talk, Eric. Thank you. I think when you were discussing ushers and time to get an outcome, it's good to look at what were the outcome failures. I mean, I look at, for example, the Mira GTX, that likely the vector that was a truncated large part of the ORF15 was likely not the appropriate vector to use. Perhaps also we don't know enough about the natural history of the diseases, so that we don't know when to assess the divergence from the natural history to the altered course with treatment. And I think there's been a rush. I think the RP65, the great success early, so you can shoot this into the eye and get an outcome immediately. And I think that it's sometimes revisiting the failures may also be appropriate to see how direct the future therapies.
Absolutely. What Gus is referring to is there was a recent phase 3 clinical trial of gene therapy for RPGR-associated retinal degeneration. The subjects in that trial are doing beautifully by most measures. Unfortunately, the primary endpoint that was pre-specified, which was again a new kind of mobility test, did not meet primary endpoints because it was variable. So I think you're totally right. We need to be informed by that. We need to make sure we're picking primary endpoints that we have confidence in and are robust. Mira GTX made a change in endpoints between phase 2 and phase 3 that probably was unfortunate choice. So absolutely, we've had some, I'm not going to call them failures. I think in many cases, I think the RPGR gene therapy works beautifully. The EMA is likely to approve that based on the secondary endpoints. But yes, we need to learn from that experience to inform our ability to do successful trials going forward, for sure.
Eric, why did Editas choose not to continue this study, given that the CRISPR-mediated therapy for CEP290 was looking promising?
You know, it's hard to know for sure what the reality is. When I've sat with Gilmore O'Neill, who's the current CEO, and asked him that question point blank, he always answers it was business reasons, right? We're in kind of a biotech winter. Venture investment in biotech and pharma is quite low at present. So companies are looking for therapies that have a high return on investment. And across the board, rare disease has become less popular as a drug development area, at least in my experience. So I think that's one of the driving forces why Editas suspended enrollment. I think another is they had expectations that were simply too high. They were looking for a miracle, for people to walk out of treatment and drive home. And the story, as we all know, is much more subtle than that. And I think they made a premature decision based on, they didn't wait long enough to see that this is actually becoming a bit of a miracle because they wanted a faster outcome in return.
Might there be a mechanism to carry it forward without them?
We're hoping so. I never wanted to found a company. I like to do, in fact, I want to go back to the lab and do bench work and experiments on this kind of stuff. But we have founded a small biotech and we're negotiating that license with Editas to be able to move this forward.
Art.
Going back to the EVs, would you know, or is it known, the turnover rate of the EVs in the plasma? And I'm thinking, you know, the obvious directions of treatments, some of which cause very quick responses and quick improvements, as opposed to, you know, watching patients for 3-4 years. In terms of applicability of this readout, do we know anything about the turnover rate?
You know, I don't. Jeff, can you help there?
We don't really know much at all about EV turnovers or EV kinetics. Certainly in ocular fluid, plasma is a very complex fluid compared to ocular fluid. So the field is still very much working.
Thanks.
We have time for one more question. Okay, thank you, Eric.
Great. Thank you all very much.